Let's start again. This is session 5, Other Models for T-cell Activation and Tolerance, part 2. The next speaker will be Zlatko Dembic. Zlatko is professor at the Faculty of Dentistry, next building. I could say a lot about Zlatko. First, maybe that he's probably one of the oldest friends of Polly here in the room, together with Peter. Zlatko has been at the Basel Institute, that's where... Zlatko is
since 83.
He's doing many important
84.
Contributions. He's in particular associate editor in the Scandinavian Journal of Immunology, and actively I started, we've heard at the initiative maybe of Polly, this discussion forum in the Scandinavian Journal of Immunology, which is really a unique place in the scientific literature for discussion, opinion articles about immunology models. So I can really recommend you to follow this discussion forum and to contribute, not the least. Just send articles. Click on Zlatko Dembic as associate editor, and your manuscript is likely to be reviewed by people like Colin Anderson, Malcolm, and so on. So it will be really well reviewed and published at the end, isn't it? Zlatko is really good at promoting discussion. So now I will stop the introduction here. I'm very happy to have Zlatko here to tell us about his model, because he has also a model, and it's called, the title of the talk is On Integrity and Immunity. You're very much welcome, Zlatko.
Thank you very much, Alex. I thank the organizers for inviting me here. I thank all my friends and colleagues for keeping and listening to me. The reason why I have a model, as you heard, is because of Polly. Polly and many of colleagues at the Basel Institute of Immunology started to discuss about things, and it was Polly that we have to thank, that she kind of distracted us from the dogma that was at that time self-non-self discrimination. Why was that important aspect? To me, it was like something that you don't ask. Of course, you know, you have a specificity of lymphocytes, and you kind of intuitively learn that you would not react to your own antigens, but you would need this system to detect, recognize, dangerous, pathogenic, non-self antigens in the environment. The reason why self, non-self was in use was obvious, because we need to have a repertoire to detect microorganisms that could cause illness or make us sick, and therefore you have to, get rid of those that could harm you. That was simple. However, there was something else, and that else was this dirty little trick of immunologists, and that was the complete Freud adjuvants, by, as Janeway noted, that actually was in the definition of the immune response in experimental systems, where without it you would not normally get good immune response defined by T and B cells. And thus it was first Janeway that came with pathogenicity, pattern or hallmark, and later on Pauly took it to the level that now we have here and in the world, that there is danger and dangerous aspect to be reckoned with. And this is why I thank Polly for introducing it, and I wish to help. However, the reason why I became editor in the Scandinavian Journal of Immunology was because I reviewed Mel Cohn's paper, which was a critique on Polly's danger. Because Mel Cohn said, well, if all lymphocytes or the cells would wave a flag of danger, 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 then the microorganisms or bacteria or viruses would mimic that danger and somehow evolutionarily will get rid of it and somehow, overcome that danger. So it should not be the reason that generates the repertoire of diversity in our systems. So I figured out that I might know the answer, but not being at the same place and working at the same place as Polly, I used my imagination. What would Polly say? Now, now here comes Mel and said, no, this is your idea. You have your own theory. And somehow I thought Mel was saying, well, divide and conquer, divide et impera, meaning, okay, the more hypotheses you have, the easier would be for me to get back into the groove and, have the dogma still defended, which turned out luckily not to be the case because slowly the danger is coming up. Why my theory is called integrity is because it is based on the giants. Like Polly said, her theory is based on the previous theories, and so mine is based on Polly's theory, but I cannot say in total because at a certain point I have my own explanation and she would maybe disagree. So here is the time and place that I could point out some of those differences. Whether they are minor or major, it doesn't matter, but it might bring up perhaps some new experimentation in the future that might open new ways for new therapies and the benefits for the society. So why integrity? Well, I picked up on the old Mechnikov idea that internal milieu need to be fully functional, operational, and therefore it has to be integrated as in organ that Peter Brecher actually is now claiming in his second book. So the immune system is viewed in this model as functionally integrated organ. It is spread among tissues of an organism, but unlike the nervous system, it migrates. So here is my point. I believe that even though we are talking about signal 1, signal 2, signal 3, which are digital in this digital era, actually in vivo they are all analog. So the signal 1 represents the accumulation of certain analog signaling among the cells in an organism and among the cells of the immune system, both innate or adaptive. So its communications are affected by cell-cell interactions and cytokines. These cytokines are interleukins, chemokines. So I assign to immunocytes location, alert status, and role. Now, nowadays we have CD cluster of differentiation molecules up to 500, 700, how many there are, I don't know. And if we just say a CD4 T cell or CD8 T cell that has other markers, we don't know these things, but we are trying to assign location, alert status, and role. Location might be, well, central organs or lymph nodes or other parts of the tissues of somatic anywhere in the organism. Alert status is the one that I would like to assign as influenced by the danger, integrity, or pathogenicity recognizing features, meaning that there is DEFCON 1 to DEFCON 4, or maybe I didn't get that right. Maybe, should I say, so naive, quiescent cells, then activated, effector cells. So this is the alert status. And maybe there are some allergic cells, and maybe which we don't know whether they are really allergic or on the way to die or whatever. So there are some, there is a space here for which I believe I have a little bit better explanation. The role is actually the class of the immune response, meaning T helper 1, T helper 2, Th17, and of course in the B cell different immunoglobulin producing cells. Now this has been described, and some of the things that I'm going to talk in my book published in July 2015, and it's about the role of cytokines in disease related to the immune response, where here are the participants. So I have the autonomous immunity and central immunity. Why? As I said, I started to talk about the comparison to the nervous system, and I believe that the immune system has a way to think. Now please don't smile or laugh too much. Subconsciously, we actually feel our own tissues. So I believe that the immune system is feeling the integrity of our organism, and they communicate, and these signals are being delivered. And that network has not been tapped yet. And this part is, on a constant on in the normal living organism. So if there is a break in this integrity signal, then there is a way for the immune system to note that. How? Well, here are the tissues. We all know about the immature and mature dendritic cells that via afferent route go to the lymph nodes, which are the central places where the signal is being transmitted via T cells and via then T to B cells, becoming effector T or effector B cells, which differentiate into plasma cells, and in the efferent route, they go back to the tissues and do the work and the job. So we see that in this case, the means for communication is the repertoire of T and B cells, so that they can discern what is happening in these tissues. Now, T cells and B cells develop. In the thymus, the T cell development goes via the hematopoietic progenitor from bone marrow via several stages. Gamma delta T cells develop first and go out with the gamma delta receptor, whereas the CD4 and CD8 actually go via double positive stages where they are clonally deleted and positively selected and go into thymus medulla and then go out. I don't know why I picked this slide, but because I like it, maybe because the next set of slides are pretty boring. So here, but there will be some pretty pictures at the end, so I promise. So here comes the major thing. So there are three main questions that the immune system should answer: to respond or not, by either defense or asylum function, to delay or not the response, and which type to evoke for executing and memorizing the response. Now, the question one is guided by the integrity principle modified by the need for defense. I state that the integrity principle is a driving force in parallel to evolutionary selection principle, which drives two biologic organisms to be together. And they actually try to, I call it sometimes symbiosis drive. And that might be the reason why we have commensals. Because I will put it at an equal footing as the negative selection. Therefore, the function of the immune system might not be only defensive, even though by definition it says so. So here is my reasoning. So let us assume that under such principle, a preferential binding exists. Let's call it a potential for commensalism between the host and the organism invading the host. This state needs a reaction from the host, and we will call it a response. For it, host needs information about the behavior of the invader. If insiders, that means secret, not really knowing what it wants to do, not clear whether the invading organism would pose a threat to host survival, the response will be delayed. And we come here to the second question. But before I go further, the evolutionary selective force here is thought to be the need to assemble or cooperate with others for a potential gain of some hitherto unknown characteristic or function that is, in retrospect, was of selective advantage. For example, spending less energy in accessing life resources at that particular time. We might come back to that in the discussion. So the next question is also guided by the same principle, and this is the delay is required when defense is obviously not immediately needed, and the loss of potential commensalism could be more disadvantageous than the potential threat of infection and death. If delayed, then the measuring of pros and cons for commensalism versus infection damage is taking place in tissues. The signals are multiple, and result must be integrated by the cells of the immune system. Upon integration, a decision to sway the immune response is held by the tissues where the immune cells are directed to. As soon as it is done, the last question needs to be answered, and that is divided in two branches. First, if defense will be selected, then the question remains, which type of the rejection response should be used? For example, Th1, Th2, Th17. If the asylum function will develop, that is, protection of commensal bacteria, what kind of effectors should be employed, like Th3 or Tregs? Other models that we had today do not mention delay. The reason for overlooking the question 2 is perhaps that in clear cases, resolution of question 1 for defense purposes exceeds by far the need for asylum function in every day. Now, here is something what Alex was trying to draw today, but maybe it is in my book. Anyway, this is how I see the self-non-self discrimination model, and it is the pattern recognition model by Janeway and Medjetov, where here you have, and I guess, Polly, you know that because similar things I was published 15 years back. So the APC would, by pattern recognition receptor, recognize a microbe, let's say pathogenic microbe, and that would give intercellular signal to upregulate co-stimulatory molecules. And in first place, it will be like digested, then presented on an MHC class 2 peptides. And this would be signal 1. The signal 2 would be co-stimulation via CD8086, and a receptor would be CD28 for that. Now the danger model, based on this, you have these two signals where Polly now says that she also accepts the co-inhibition or the balance of the co-stimulation and co-inhibition, which I think it is fair to say that has been contributed by Sinclair and Anderson. Now here we have the danger signal or signal 0, which is the outside signal from the tissues on the APC. So here the antigenic peptide is signal 1, tissue debris, so necrotic cell death, tissue distress or damage, the 3Ds, are the signal 0. Well, here we come to the integrity model. It's divided in 2 slides. First, let's take a look what is the APC naive T cell. So let's say the APCs have signals which are transferred via pattern recognition receptors or the intracellular pattern, and there are other receptors that could detect the patterns, the stress signals, inflammasomes, and so on. There is a suboptimal signal in TLRs, and I don't want to go in detail of it, but basically TLR2 and TLR9 suboptimal signaling can be integrated. So I assume or suggest that there are 2 signals from the pattern recognition receptor. So if there is suboptimal signal from one of the receptor outside and there is another one from inside, that would yield to or be equal to the activation or co-stimulation. So here we have these 2 signals, but I suggest that there is a third signal, the damage from the harmful microbes, which induces modulation of APC's activation. This opens room for the discussion of how these APCs in the periphery can be activated. Basically, I suggest that there are 3 signals that guide the development or activation or maturation of dendritic cells, for example, that might migrate to lymph node to give these signals to the naive T cells. Now here is the difference. I propose that this integrity change, which is putative, I don't know what it is. It could be perhaps additional T cell or perhaps just a stromal cell somewhere. So here we have this possibility to have this communication with other cells of the immune system. If this is affected by the effector T cell with different specificity, it would be like the 2 signal from Peter Brecher. And the danger, the difference between the danger is here that I suppose that there are intracellular and extracellular, and both need to work on the antigen-presenting cells, whereas the danger is only from the outside, as you were saying. Pathogenicity signal would be from the inside of the APCs. I suggest that the lack of integrity signaling would say, oh yeah, there is something wrong, basically, with the tissue. But it would need additionally the danger signal. So the I suggest there are integrity-associated molecular patterns that need to be recognized in addition to the danger-associated pattern or dumps or paths. Okay, so this is an example of the fence case where harmful microbes invade the host. For vigilance, suboptimal signals can prime the APCs. Now, there is something else that I will come and address at the end, which is not far away. So here are the toll-like receptors and the ligands. Just for the sake, there's another nice slide, and I hope that you are not bored with all these texts. So here, the reason why I put it, because only recently we saw that there is only one which is anti-inflammatory, and that's the TLR10, which actually takes TLR2 out of these two heterodimeric parts which are pro-inflammatory. So there is, even at the pattern recognition receptor, a balance between pro-inflammatory and anti-inflammatory. Now here comes the second part of the integrity model. How do B cells get activated? So naive B cell or virgin B cell gets a signal 1 from the antigen of the harmful microbe, let's say, and the T cell effector that is specific for the peptide or hapten that is carried here. So that's help. And here is the caveat. We need an integrity change additional signal 3, which I don't know whether it is another T cell or a B cell or stromal cell. So the signal 3 transmits, in this case, the damage caused by the harmful microbes. The integrity model needs to address also the role of the MHC. So the MHC developed from its role as a sentinel post for environmental antigens. So it would be like a receptor for the antigens. It had the alarm functioning in detecting, yes, dangerous microorganisms. It has genetically expanded for that purpose in higher vertebrates. Based on that premise, defense dominates the decision-making processes defined as interactions between immune cells and tissues nowadays, and therefore the asylum function, commensalism, symbiosis drive, has been neglected in our conceptual maps. I suggest that the defensive sentinel function of the MHC piggybacked the integrity principle through phylogeny, now hidden due to the change in environmental complexity. For example, in other words, an organism contacts pathogenic parasitic environment and its antigens much more frequently now than in historical times, and defense is taking precedence over symbiotic drive function of the immune system. For example, some animals, mole rats, that live underground might have simpler environments, and hence their defensive immune reactions might be downsized to the level that they do not need many MHC molecules to tap the antigenic landscape of the environment for defensive purposes. If we accept the integrity principle, then we should put asylum function as a principle on equal footing with defense principle in deciding how the immune system would respond. The next thing is, if defense has memory, then can an asylum seeker be memorized too? My answer would be affirmative and predicted by the asylum function. Beneficial commensals are remembered, but hold in check, as mutations could diversify them into pathogens and parasites. Thus, we are coming to the last function, which is a hallmark characteristic of the immune system, which is vigilance. The immune system has to be constantly on alert and exercise vigilance because the mutual benefit may abruptly end with the commensals. It may be envisaged that sudden change in conditions of the environment would lead to competition for food, energy, and other resources, and this event would surpass the potential gains for commensalism. Thus, the constant monitoring of signals from tissues, from immediate environment, gives information to the immune system. This vigilance is the readiness of T and B cells to perform the three-question algorithm that we started with. Their corporation makes a decision based on the answer to the 3 questions by cellular contacts and cytokine signaling. So here is the summary of outcomes. So if there is no damage as a signal 3, the DTCs would be vigilant or be energized. So that there would be a neglection of neutral microbes, which would be the explanation for energy. Now, if there are harmful microbes, they would be combated by T effectors and B effectors, so that the signal 3, there is a damage defect detected, the T cells would become effectors. There would be a positive feedback that would increase this response, and they would then transfer the signal to the B cells, which would be expanded and used in combat. On the other side is the asylum function or protection, where if there is no damage and useful signals are, which I don't know which they are, but useful integrity signals, maybe vitamin K production, you would have a negative feedback by Tregs or negative feedback by Bregs here, and these would be like a military police. They would protect the commensals by actually downsizing the combat function of the immune system. So what would be the prediction? And this is the summary, I guess, one before the last slide. So for the host homeostasis, in defense function, you have a TMB repertoire that is clonally deleted in the thymus or bone marrow, called self-tolerance, and in the periphery for membrane-bound self-antigens. These are things that have been described and discussed. The vigilance function would make peripheral tolerance actually energy of self-tissues for soluble self-antigens. So this means that if there is a third signal saying that there is danger coming up, they might be reactivated. And the third is the protection of self-integrity by Tregs and interplay of TH-type switching. Why I'm saying this? I say that Tregs basically have additional function in keeping the integrity of the whole organism. Now, in the affection, you have combating harmful microbes and parasites by T and B effectors, and that's the normal immune response that we know. Here, in the vigilance, the neglection of neutral microbes, that's the reason for the delay of the response by energy of effect or by energy of effectors. The integrity function or hallmark during the infection would be that it might give asylum to useful microbes by Tregs. In the autoimmune disease, you would have a failure of clonal deletion in thymus or bone marrow and periphery, and these autoreactive T and B immunocytes, as we discussed and as everybody pointed out, that they would cause the if they are cross-reactive to foreign antigens, they would give autoimmunity. Here, the neglection would fail. Allergic cells might become activated by the failure of the tissue that is actually being attacked then. Here, Tregs fail to become generated, or there is a failure in the TH switch. In the cancer, you have mutating antigens to escape immunity or clonally delete thymocytes, T and B immunocytes. Here, the cancer would fake neutrality to delay decision or energize immune cell effectors. And in the last case, faking integrity signals to engage Tregs or causing TH shift, thus evading immune attack. Now, if we have two choices to discriminate between danger and non-danger, or self and non-self, and if we have three choices, like I'm suggesting, then how would the world look like? And the self-non-self discrimination, or any other two-choice discrimination, you would have a rejection or tolerance, which is self or non-dangerous. But in the integrity model, only the harmful in red will be rejected by the immune defense. Non-harmful will be recognized by vigilance, so recognized and taken care of. But the useful in blue, tolerated and protected by the immune asylum, by example for Tregs, they might be kept and constantly be aware whether they will not turn out to be harmful. So I thank you for your vigilance and for your tolerance. And now I would like to discuss this.
Thank you very much. Thank you very much, Zlatko. Wonderful. You didn't mention I think the last slide was published in a Scandinavian journal paper.
Yes.
Entitled, what was the title of the paper?
I forgot.
50,
Remind me.
wasn't it the 50 Shades of...
Oh, no. Something like with the shades of, yeah. But yes, but it introduced that the gray is non-harmful.
That's another important paper published in a Scandinavian journal. Yes, questions or comments to Zlatko's model? Colin?
So would you go as far as saying Tregs are an integrity sentinel, like a dendritic cell is a sentinel for danger? Is the Treg cell, you talked about it, could it be a B cell, a T cell, or a stromal cell? Do you see the Treg cell as the cell that detects integrity?
Let me think. There are so many different types of Tregs being found, and there is also a TH shift that would cause similar things. So I put in point just the FOXP3, CD4, CD25 Treg as they are. I don't know. There are other CD or IL-35 Tregs, IL-17 Tregs, and there are so many, and TH3, after all, they all secrete. I think it's the interplay of all this, so no, I wouldn't point to that one of these specific Tregs to be the sentinels. But because they might be evolutionarily not selected. So I think it's the principle that they exist as such is being driven by the need to cooperate and to find a cell to associate with and actually to see the benefit, whether it is energy cost or something. I actually elaborated much more that, and I will put it in writing. I came up with formula, but I thought it was stupid. But basically, I thought that like the gravity is a force that is affected on any particle that has a mass. So I think the preference or attraction is affected in biologic sense. And that is the symbiotic drive that I'm trying to point out. Maybe the force decreases with the distance, genetic distance. So the microorganism and the eukaryotic organism might have a larger drive, but that is something that I have to elaborate more. Yes?
So I'm a little confused. If something is useful, then its presence is maintained by Tregs.
Yeah.
And if something is
harmful?
not necessarily useful, but not harmful, then its presence is maintained by deletion or energy.
Yeah.
That's how I understood the last slide. Okay, so I have a couple questions on that. First, how do you define useful? How does the Treg population know that something is useful? So that's the first question.
Okay, I'm sorry. Please, it is better that you remember your next and I answer immediately that. Can it?
Yeah.
I don't know exactly, but I would like to think that there is a cooperation, like in the nervous system, a subconscious decision between, let's say, dendritic cell, T cell, another T cell, B cell, that measures the signals of integrity. So if there is a damage plus danger in the tissues they survey, then they would activate the defense mechanism. If there is not an integrity, but if there is a beneficial thing, and that might be measured by the, for vitamin K, I was trying to think, what would be the mechanism for that usefulness? Of course, we can get vitamin K from other food sources, but in that particular, there must be something that is not necessarily just a single receptor. It has to be a network of signals that would provide usefulness, and I don't know. That's the reason why I said that I don't know. But for each usefulness, actually, it's like doing research. You don't know what you are going to detect. So the organism is constantly searching among all these signals, trying to deselect the dangerous ones, and once they are not dangerous, so it is selecting or integrating all that is useful. So if certain aspects of livelihood or functioning of the whole organism is being better, these integrated signal would be useful and is taken as useful, and this would be at the subconscious level transduced or transferred to the immunocytes via cytokines, and then sent to the location via chemokines.
But something like vitamin K production by bacteria in the gut has its usefulness in
blood,
clotting in blood, right? So the location is not the same.
No, of course. I don't think that the location of the effectors, so the Treg effectors, is guided by in the afferent loop. So that's why I said there is afferent loop and the efferent loop. So the Tregs would be given a location for the efferent loop. But yes, that is, I see the problem what you are saying. If the usefulness is detected in the whole organism, how would a Treg know where to go and to protect? That, I assume, might come from the detection by constant vigilance. So the spy network actually would tell they are there. And there might be a tag saying, okay, there this thing has been produced, and therefore we tag the effector cell to that location.
So you could do an experiment.
Yeah.
Yeah, you could either give animals bacteria that make vitamin K,
Yeah.
or you could give them a little bit of a suppository under the skin that releases vitamin K.
Yeah, for example.
And now ask if your Tregs end up in a different place.
Yeah.
Okay.
Thanks.
Second question. So it looks to me like your last slide.
Yeah.
The Tregs are there for things that are useful but not self.
Yeah.
So it seems to me
But it
that there's also an awful lot of self that is useful, right? I mean, we have kidneys that filter blood, we have a heart that beats, we have lungs that breathe. So why do you separate them that way?
I don't actually. But it might be that there are different subpopulation of Tregs.
Sure.
And therefore, here, I just wanted to say that perhaps for different organs, different Treg species are important for maintaining the integrity and ensuing the last bit of tolerance that the central and peripheral tolerance failed to do.
So, you know the paper of Chateauneuf and Bach, where they looked at 3 different populations of suppressor cells that were able to suppress autoimmunity in 3 different organs. And if you don't know that paper, you should go find it.
Okay.
So one more.
Yeah.
And this is a really basic evolution selection argument, and I'm not perfect in evolution science. But I think there are people like Richard Dawkins, for example, who would argue that when you have commensalism, it's not that there's a drive to commensalism. It's that for each organism that is part of that pact, it's better for them to be in it than for not to be in it. And that it's really, in the end, a purely selfish decision.
It's a very selfish argument, I agree. As well as his model, I respect his selfish gene hypothesis, and I like it, but not 100%. And in this aspect, I beg to differ, because we still don't know what drives the diversity, because we know that it's been selected, that because of the diversity of life, we have natural selection. But why is it? Is it because it's...
Well, it's not because of the diversity of life that we have natural selection, right? Isn't it the other way around?
Well, we don't have life on every planet or anywhere else in galaxies so far.
We don't know that.
And we don't know that, but so far we don't have any evidence. But on the other hand, I would believe, I look for a set. But basically, I think there is that life in itself is a perpetuum mobile of third kind, which means that through evolution it kind of replenishes and avoids death and avoids total loss of energy. So is that a drive, or is that just a thing that our physics or our world is based? So I believe it is a drive. So that's my viewpoint.
Okay, let's now we
Alex.
need to make a break for the lunch, but you have possibility to interact with the speaker. As you can see, there's still many unresolved issues how the immune system deals with command cells. Tregs, that was basically the first attempt we've heard at this meeting to incorporate Tregs in models. Thank you very much, Zlatko. And welcome back, everybody. So there is some lunch served outside, and welcome back at 1:15.